He has also received the Best Doctor Award in Medical Education from Kerala Government and M.M. Ghani Award of Calicut University in the year 1994 and 95 respectively. His textbook of biochemistry for medical students is being followed by Indian and foreign universities, of which Spanish and Russian editions are also available. He has also authored textbooks for dental and paramedical students. He has guided 29 students for their PhD work. There are more than 240 publications in highly acclaimed journals to his credit. After his retirement from government service, he actively served at Kasturba Medical College, both Manipal and Mangalore, and as founder principal at Sikkim Manipal University and Amrita Medical College, Kochi. He continues to share his expertise with Amrita and Agape Diagnoscs, Kochi. Dr. Vasudevan was the founder director of Research Institute at RCC Trivandrum as well as Amala Thrissur. His sustained and exceptionally high impact leadership achievements demonstrates the effectiveness in advancing the JCMR's goals and image. An endowment oration was instituted in honor of 50 years of his academic career on 2019, featuring distinguished speakers from around the world. The first orator was Dr. M.V. Pillai from US. The second orator was Dr. Shyam Potilil from US. The third orator was Dr. Gagandeep Kang from Vellore. The fourth orator was Dr. Sumesh Philip Thomas from Canada. The fifth orator was Dr. Valsraj Menon from UK. The sixth oration in 2024 was delivered by Dr. Vinod Skaria from Bangalore. Welcome to the 7th discourse of Dr. D.M. Vasudevan Endowment Oration featuring Dr. Ashwin Dalal from CDFT Hyderabad.
Let's begin the program. First of all, I request the dignitaries to kindly come to the rise and take their seats. I especially Let us begin this auspicious occasion with a prayer. I request everyone to stand for the prayer.
ശീലു ചൊല്ലുവാൻ നാവിനാകേണമേ സ്നേഹമാകുന്ന ഗീതമോ എൻ്റെ കാതിനിണയാകണേ സത്യമെന്നുള്ള ശീലമോടെ ഞാൻ ശാന്തി അറിയണമേ
Kindly be seated. Ladies and gentlemen, as we gather here today to honor the legacy of Dr. D.M. Vasudevan through his endowment oration, we take a moment to remember and pay tribute to a distinguished scientist and educator, Dr. Ravindran Angathil, who left us on 27th January 2025 at the age of 69. Dr. Angathil was a dedicated researcher in genetics and cancer studies, completing his PhD under the mentorship of Dr. D.M. Vasudevan. His illustrious career spanned over 4 decades, including his tenure as Professor of Genetics, Cytogenetics Consultant, etc., and his invaluable contributions to RCC Trivandrum. A mentor to many, his legacy continues to inspire the scientific community. In honor of his memory and contributions, may I kindly request everyone to rise for a 1-minute silence. Dr. P.R. Varghese, Research Coordinator, Jubilee Center for Medical Research, to welcome the gathering.
A very good morning to one and all. Respected dignitaries, guests, faculty, staff, students, and benefactors of Jubilee. It is a honor to me to welcome all of you to this academic festival that we are celebrating for the 7th year. Dr. D. Vasudevan is our heart and soul. With his leadership, we completed a glorious decade. All these 57 years of academic career, this legendary gentleman has been living with his students, fellow researchers, and academic counterparts. Jubilee Center for Medical Research is proud to organize the 7th discourse of Dr. D.M. Vasudevan Endowment Oration today. All about him is already played in the video just now. First of all, let me welcome our research director, Vasudevan sir, who made this day possible. Welcome, sir, most lovingly and respectfully. I also welcome Mrs. Devi Vasudevan to this function. Welcome, ma'am. The orator of this function is Dr. Ashwin Dalal, Head Diagnostic Division, Center for DNA Fingerprinting and Diagnostics, Hyderabad. Dr. Ashwin Dalal is a unique blend of pediatrician, geneticist, and a basic researcher. He leads a DBT-funded rupees 100 crore national program on pediatric rare genetic disease. That is a collaborative initiative spanning 16 prestigious institutions of the country. Dr. Dalal is involved in the drafting of National Rare Disease Policy 2021. Today, Dr. Dalal will speak on Indian Genomics Landscape, where we are and what the future holds. Sir, at Jubilee, we have different faculty members interested in molecular diagnostics. Among them, Dr. Rohini Sebastian, Assistant Professor, Department of Pathology, is a peculiar person, so we are very much fond of her. Dr. Rohini recently completed her fellowship program at CMC Vellore and is an admirer of your Dr. Dalal sir. So, now it is my pleasure to welcome Dr. Ashwin Dalal sir to this program. I request Dr. Rohini to offer bouquet. Hearty welcome, sir, to this program. Thank you, Dr. Rohini. Our director, Father Renny Mundangurian, will formally inaugurate this function. Father is our strength. Welcome, Father, to this program. Dr. Pravinlal Kutichara, the principal of Jubilee Mission Medical College, is presiding over this program. He is not only an administrator, but a true researcher. Welcome, sir, to this program. I am extending a warm welcome to our CEO, Dr. Benny Joseph. He is not here. He will arrive in short time. Hearty welcome to Benny, sir. I also welcome Father Terin Mullakara, our assistant director. Even though Father is a new person to this institution, he lends a great support to all our endeavors that initiated by the research department. Welcome, Father, to this program. I welcome all academic persons from neighboring institutions, our benefactors, friends of Dr. Ashwin Dalal, And a warm welcome to all our assistant directors, vice principals, nursing superintendent, deputy nursing superintendents, and especially medical superintendent Dr. Shibu C. Kallivalapil to this program. Welcome everyone. I am happy to welcome all heads of Jubilee institutions, faculty, staff and students of Jubilee institutions to this program. A warm welcome to media persons to this program. A special welcome to all our JCMR team members who is working hard for the smooth conduct of this program. Special welcome to Dr. Alex George, who will formally introduce the speaker, and Dr. Matthew John, who will formally propose a vote of thanks. Once again, I welcome you all, wishing you a great academic feat. Welcome all.
Thank you, sir, and we welcome you for the program. Now I invite Dr. Praveen Lal Kuttichira, Principal, Jubilee Mission Medical College and Research Institute, to deliver the presidential address.
Respected dignitaries on the dais, my fellow colleagues, dear students, scientists, Gurur Brahma, Gurur Vishnu, Gurur Devo Maheshwara, Guru Sashal Parabrahma, Tasmai Sri Gurave Namaha. Words of Adi Shankara. It means teacher is the force of creation. Teacher is the force of preservation. Teacher is the force of ultimate transformation. Teacher is actually the universal eternity. I bow my head before the teacher. Years back, when K.K. Vijayan sitting before me in the front row, we were known each other from the days of Mavur Goliar Rayon's agitation against the ecological issues in that area. In PG Hostel Trivandrum, we were living in the nearby rooms. He was doing his PhD. K.K. Vijayan, myself, and Ravindran Angathil, whom we lost recently. We were friends. To meet them, I used to peep into the two-room center in the medical college, just above the radiation room, where Dr. Vasudevan had set up the laboratory. Only 2 rooms. That is the place where I saw first time in my life the automated laboratory machines. Sir was working there in charge of the research wing. The clinical side was looking after by Dr. Krishnan Nair. And from there the RCC came up now. It is from there the cancer research in Kerala reached the present status. So I witnessed the growth of it. The growth nurtured by Vasudevan sir. Soon after India became independent, in early 1950s, when 5 nations of the world sat together on the initiative of Pandit Jawaharlal Nehru, AIIMS New Delhi came up. And the biochemistry department of AIIMS New Delhi, even when the brains behind that time would have never thought of it. The Department of Biochemistry of AIMS contributed 2 persons to Kerala. One is Dr. C.R. Soman, who had done hand-holding of Kerala medical sciences to the community for the public health. Second is Dr. D.M. Vasudevan, who did hand-holding of Kerala medical sciences to the wet labs for the basic medical research. Vasudevan sir's presence in the arena is just like the multi-stage Amittu of Thrissur Puram. One burst will give rise to many other bursts at different stages. The whole Puram ground will be illuminated. That's what Sir had done. Vasudevan sir's disciples, after PhDs, dispersed to different parts of the world, different parts of the country, different elite centers in this state, all headed by Sir's students. And from where the new stages of eruptions comes up. And it gets continued even now. There is a saying, a real teacher is one, the need for whom gradually fades away. With 14 of our MBBS students now in the campus having STS scholarship, with 13 of the students with STS, 14 students with the Kerala Health Sciences University Research Appreciation Awards, again during the same year, and 3 postgraduate students getting ICMR grant for doing their thesis work. Total 30. 30 of the research minds in this campus at the moment. I would say the saying I mentioned is actualized with Dr. D.M. Vasudevan at the head of the JCMR. Sir has got unique talents. He started academic institutions, maintained it, and handed over to others. From Amrita Kochi to Amrita Sikkim Manipal, Manipal, everywhere. We know people who initiate things and leave. Their role ends there. We know some other set of people runs it, manages it. We know the most difficult task in any institution or any department is transition management. All 3 done successfully by Dr. Vasudevan. Another area is academics and industry. Both Sir is involved. Sir could initiate and maintain relationships between these 2, mutually supporting and fostering. These are the unique talents of Dr. Vasudevan. I stop here because I can't give actual picture of Dr. Vasudevan before you. You may be knowing a different part of Dr. Vasudevan. Someone else entering this hall now may have something else to describe about him. I can only say we have Dr. Vasudevan with us and we have a teacher with us. My best wishes.
Thank you so much, sir. Light symbolizes knowledge, purity, and is the dispeller of darkness. To formally inaugurate today's event, I request Reverend Father Rennie Mundangurian, Director of Jubilee Institutions, to come forward and light the ceremonial lamp. I also request other dignitaries to join in. Thank you all. I invite a director for the inaugural address.
Have a loving good morning to you all, distinguished guests on the dais and off the dais. A hearty welcome to Jubilee, especially to the 7th DM Vasudevan Memorial Oration. Endowment Oration, yeah. As you know, Jubilee is an institution of history of 73 years. 1951 started, and we have now 7 institutions under the name Jubilee. The hospital, the nursing school, medical college, nursing college, College of Allied Sciences, and the 7th is our research. 6th is the research. Yeah, otherwise Dr. Varghese will not agree. So 6th child is research, and the 7th is the Ayurveda hospital in Chero. When we take a year of programs in Jubilee, almost every day there will be a program in one or other institution of Jubilee. I have to attend. Almost every day programs are there. And I can say the first Wednesday of every year, the last 7 years, is a day of joy for me. So it is the Dr. DM Vasudevan Endowment Oration. Every year, first Wednesday of March. For 3 reasons I stay here with a joyful heart. The first is because of the name Dr. Vasudevan. I don't go to the detail. We saw some pages of his life on the monitor, and some pages described by Dr. Praveen, and I don't go to the detail. I can say Jubilee Research is a flower nurtured by Dr. Vasudevan. 10 years is for by, now 11th year, and I can say Jubilee Research is entering a 3rd stage of development. The first, the minimal infrastructure, started with minimal infrastructure. The second was to imbibe interest of research in all 7 institutions, the Jubilee families of all the 7 institutions. That was the second stage. It will be continuing also. And 3rd stage is coming without late. We are planning an expansion of the Jubilee Research Center in the 7th or the 6th or 7th floor of this building. This is 3rd expansion of the Jubilee Research. Today we had where distinguished guests are there. One among this, the principal of the Kolancheri Medical College. He spoke about the research and spoke about the invest we have to put in the research. We may not see the result immediately, but only those who can have a long vision, distant vision, they can invest in the research. And he's happy about our research and asked about the expansion. When we think about the expansion, it's a huge investment, it's a third expansion, but we will do with a serenal heart because of the presence of Dr. Vasudevan. We are confident it will be a successful expansion of the research. He's like a calm river, flows everywhere, give lives wherever it flows. And some of the pages we saw, wherever he was, there was life. And I can say Jubilee Research is a lively research center because of his presence. We are really proud of his presence. Thank you, sir, for your presence in Jubilee. Really a soothing presence, an inspiring presence. And we hope to continue that. So that's the first reason I can say today I'm standing here with a joyful heart. Some successful stories described by Dr. Praveen Lal. The faculties are coming forward, especially the last steps are the coming of the students. In all over India, research projects we are getting awards. For our MBBS students, it is also signs of the successful presence of Dr. Vasudevan. So we are really proud of it. And I thought to describe the last 6 orations, but it was presented in the monitor, so I don't go to the details. Today is the 7th oration, and it is an academic feast. I have to say, every year, first Wednesday of March is a day of academic feast. Every year, the oration was selected really after a long process of pondering, meditation, and every year when we get what we get is the best in this field. We have a good advisory board. Some of the advisory board members are present here. They evaluate and suggest, always concentrate 2 or 3 areas for the research. One of their suggestion was also genomics, and this year it is going deeply, and I'm happy Dr. Ashwin Dalal, we got luckily eminent person. I don't got the detail, it will be followed without late. Dr. Alex will present Dr. Ashwin, and we are really happy to have him amongst us. Sir, thank you very much for coming to us. And welcome you all to this academic feast, and thank you for your presence. As told by Dr. Varghese, with your permission, I declare the 7th Dr. Vasudevan Endowment Oration 2025 is inaugurated. Thank you very much.
Thank you, Father. Now I invite Dr. Alex George, Scientist J.C. Marr, to introduce our esteemed orator of the day. Over to you, sir.
Good morning. Today, I am honored to present Dr. Ashwin Dalal, a pioneer in medical genetics and the chief scientist at the Center for DNA Fingerprinting and Diagnostics. His work has significantly advanced genetic diagnostics and rare disease research in India. Dr. Dalal's journey started with passion for science and medicine. Born on December 4th, 1975, he excelled academically, earning a gold medal in his MBBS exams. His early interest in genetics and patient care laid the foundation for his illustrious career. He completed his schooling at Jawahar Navodaya Vidyalaya, Goa, demonstrating academic excellence from an early age. He then pursued an MBBS with distinction, followed by an MD in pediatrics from Goa University, Further specializing in genetics, he obtained a DEM Medical Genetics from SGPG IMS, Lucknow. His educational journey reflects a profound dedication to understanding genetic disorders and their implications for human health. After serving as a senior resident in pediatrics, he joined CDFD in 2007. He later became head of the Diagnostics division and also played a key role in establishing the medical genetics department at NIMS Hyderabad. His contributions to academia include serving as a visiting professor at Kasturba Medical College. With over 170 research publications, Dr. Dalal has made groundbreaking discoveries in medical genetics. He has identified novel genes linked to rare disorders, including BHLHA9, AIMP2, ARMC9, and SERPIN11. His work on genetic variants has improved disease diagnosis and treatment approaches. He also played a vital role in sequencing COVID-19 samples as part of the INSACOG consortium. As chief scientist and head of the diagnostic division at CDFD, he leads a state-of-the-art genetic diagnostics lab. He specializes in prenatal screening and rare disease diagnosis, offering genetic counseling to thousands of families. His expertise has helped assess over 60,000 patients. His DBT-funded program on pediatric rare disease collaborates with 16 institutes utilizing genome sequencing for novel gene identification. He established the National Genomics Core to advance genomics research. Additionally, he has contributed to India's National Rare Disease Policy 2021 and developed genetic database aiding diagnostics. He serves on key government task forces guiding genetic research and healthcare initiatives. As a mentor, he has trained numerous clinicians and scientists, ensuring the growth of medical genetics in India. Dr. Dalal continues to push the boundaries of genetic diagnosis and personalized medicine. His vision includes expanding genetic services to underserved communities, bridging research with clinical applications, and enhancing precision medicine for improved healthcare outcomes. In summary, Dr. Ashwin Dalal is a leader in medical genetics, transforming patient care through research, diagnostics, and policy contributions. His work has laid the foundation for future advancements in genomic medicine, making a lasting impact on India's healthcare landscape. With this, I conclude in introducing him. Thank you.
Thank you, sir. Now it's time for the much-awaited oration. I request other dignitaries to kindly join the audience. Ladies and gentlemen, let us welcome Dr. Ashwin Dalal to deliver oration lecture.
You can handle this. Videos are there, so mouse will be fine for you. To click. Can you just scroll this zone, or are you fine with that? Yeah, I can scroll. This you can point out the point. It will make a dark mode into it. Next is will be here. Okay. Next is here. This is the pointer. Pointer won't work. You can use this, I think. The next is this one. With the arrows also it will work? Yeah, it will work. You can change it like this also. Okay. You just hold it like this and it will work. Oh, okay. Fine. This is the next. This is the previous. Bottom. Fine.
A very good morning to all of you, and thank you Dr. Alex for that introduction, and thank you Dr. Varghese and Jubilee Medical Research Center for considering me worth to deliver the Dr. D.M. Vasudevan Endowment Oration for this year. And I hope at the end of my talk I am able to inspire at least few of you to jump into this field of genomics and do much more than what we have achieved till now. So to talk about the Indian genomics landscape, I thought I'll use my first part of the talk to introduce to you to the field of genomics through 4 stories that I have put together, and then in the next part we'll try and see what is happening in the field of genomics, what we have been doing as part of our research in the field of genomics, and we'll see where we can go from here. So to begin with, if you look at our country India, it's really been a melting pot of cultures. We have had so many different cultures, so many different origins of people. We have the Austroasiatic, we have the Indo-Aryan, we have the Dravidian, and a lot of endogamous groups. So from all these things, from the genetics or the genomics point of view, it becomes a gold mine for doing research, and at the same time it becomes a problem also, because we have such a huge population and hence we have such a huge load of rare diseases. Even if we call them rare, there are more than 7 crore patients with rare disease in India. So you really can't call them as rare diseases. So it's a huge burden that we need to tackle with, and that is why there is a need for a lot of research and initiatives in this field. So as I said, let us delve into this area by going through 4 stories that I would like to share you and to introduce to this field using the 4 letters that form the crux of human genome, that is A, T, G and C. Okay? So let's start with the letter A, which codes for adenine in our DNA, and I term it as a book of life. So A represents a book of life. So if you look at the different books that are written in English language, then all the millions and millions of books would be written using this 26 alphabets. That's the code of the of the English language. Similarly, our genome, which is a 3 billion long stretch of DNA, it uses just this 4 nucleotide A, T, G and C. Instead of the 26 alphabets, it uses the 4 alphabets A, T, G and C. And these 4 alphabets in different combinations lead to our 3 billion genome. And that just like you can write so many different types of books using these letters, in the same way, all the different human individuals on this earth, all different from each other, are all coming from different combinations of this A, T, G and C. And just like where you write your English language, you can make some mistakes. Our genome also makes some mistake, but not forgiven. And one change in 3 billion, and you can get a severe disease like sickle cell anemia, a very common disease. So even one change in 3 billion is not accepted by the genome, and that is where the importance of understanding the genome comes to make treatments or diagnostics for these diseases. Now, how do we go about finding these mistakes? So if you look from the book point of view, if you have the first edition of the book and you know where is the mistake, you go to the second edition of the book. On that same page you can look for whether that mistake is there or not there. And same way, if we know a particular mutation, say sickle cell disease, if we know what is the disease, we know what is the mutation, in the second patient we can look for the same mutation, and for that we use a technique called as Sanger sequencing. This is the technique that was developed to sequence the human genome. The whole human genome was sequenced using this technique. A very simple technique, a very visually interpretable technique. You can nicely see the peaks of A, T, G and C and check whether the patient has the mutation or not. Okay? So this is much easy, but when you want to find an unknown mutation, when you don't know what is the mutation, what is the disease that you are dealing with, patient has come with intellectual disability or some malformations. Here finding mutation is like finding this dot in the whole Encyclopedia Britannica, because we are trying to find 1 in 3 billion. Okay? So that's the amount of complexity our genome holds and the difficulty in identifying these mutations. And it's really like finding needle in a haystack. Okay? And so if you want to find needle in a haystack, you are not really going to be successful unless you do a large amount of sequencing. And so we have the technologies now after Sanger sequencing. Now what we have is something called as a exome sequencing or a whole genome sequencing. So the technology now enables us to sequence either the whole genome of 3 billion base pairs, or you can sequence only the coding part of the human DNA, which is only 1% of the human DNA that is coding, but it is responsible for more than 85% of the mutations in genetic diseases. So it makes sense to sequence just this 1% and detect all those 85% of patients with genetic diseases. And that is why exome sequencing is what we use routinely in our clinical practice. But when you don't find something on an exome, you will go to something called a whole genome sequencing. But when you sequence such huge amount, the exome is 30 million base pairs, the genome is 3 billion base pairs. When you sequence, you can't go like this finding the needle in the haystack. You will have to systematically collect the hay, break it into small groups, take one group at a time, you will find different needles, use some sort of sieve, and then you reach to that needle. And that is what we do in our bioinformatic analysis pipeline. So today's clinician, today's basic researcher has to have this dual expertise of your wet lab research as well as the bioinformatic research. We cannot say that I am a wet lab researcher, I don't need any computer knowledge or bioinformatics. The amount of data that comes out of our genome, proteome, transcriptome, these are huge amount of data, and unless we are trained in the use of computers, we really cannot do good research. So it is very important for today's students who are aspiring either to be a clinician or a basic researcher to make sure that you are aware of the computational aspects of biology. Now, because of all this huge amount of data, we really have a data deluge. Okay, exome generates about 5 GB. All of you must be aware about the GBs and the MBs because we all use computers. We know the size. So same way, when you count the exome, it's 5 GB data that comes out of each. You have 25,000 variants that come out which need to be analyzed. So a lot of high power computing is needed, a lot of computer storage is needed. And now think, when we start doing whole genomes, it's typically 90 GB of data that comes out of one genome. And now think, if you start doing thousands of genomes, it's a huge amount of storage space, huge servers are needed. And with this huge amount of data comes a great power to us, because our genome holds the key to everything what we are today and what we are going to be in future. The genome holds the key to what diseases we are predisposed to what drugs that we will respond to. And so this huge power we have in our hand because of the availability of these powerful techniques of genome sequencing. However, when you get great powers, you also have to remember to use it with responsibility. Okay? And so with great powers of these tools, we also have a lot of ethical, social and legal issues that we need to keep in mind. We have so many incidental findings. We are looking for a point mutation for a particular disease. We find that that patient has something else because now we have the whole genome available to us. If you find out that a person is predisposed, are we going to discriminate that person in insurance or in employment? How do we return these findings? All these issues come up when you have this large amount of data. We use typically exome and genome sequencing for identifying novel genes. That's the area of research that I work on. So I'll take you through some of our endeavors, how we went about analyzing the exomes and genomes to identify these novel genes. Because why we need to identify novel genes is because we have about 19,500 genes in each of our cells. Our genome codes for 19,500 genes roughly, but we know diseases only for about 7,000. So more than 12,000 genes are still remaining to be identified what happens when there is a mutation in those genes. So a lot of work remains to be done in this field. And one such example is this patient. So this was a consanguineous family that we had. And the first patient, it was the patient was seen by my guide, Dr. Shubha Phadke, in 1999. And you can see the abnormalities in the hands and the feet here. So there were multiple abnormalities in the hands. And then this patient, this family came for the next pregnancy, and the fetus also had the same abnormality. And so this was looking like an autosomal recessive disorder. But back in 1999, we really did not have these tools of exome or genome sequencing. However, in 2011 and 12, when the next generation sequencing came and we were able to do these exome sequencings, we traced the patient back, and now the patient was an adult. You can see that the patient has undergone multiple plastic surgeries to get those extra fingers removed. And we thought that why not use this technology to see whether we can understand what is the gene responsible for causing this abnormality. So that time it was just called as complex camptopolydactyly, and it was enlisted in OMIM, that is online Mendelian inheritance in man. So when we called this patient back and we got the exome sequencing done, this is what happens. You get about 27,000 variants in the exome. So these variants are differences among people. We all are different from each other because we all are same at 99.9 positions in the genome. 0.1% difference in the genome makes us all different from each other. But 0.1% of genome, you can imagine, is millions of base pairs. Okay? So when you do an exome, you typically get about 27, 28, 30,000 variants. And then we use different criteria to filter those variants. Ultimately, we could come down to one particular variant in a gene. What Dr. Alex also told you was BHLH-A9 gene. So this BHLH-A9 gene was a gene, nothing was known about this gene, what it causes. We thought probably this particular change, you can see that the patient, the normal is GA, and our patient is having a TT in that position. So we thought we should see whether this gene is really the cause of disease in this patient. So we always do Sanger sequencing to confirm, and you can see here the normal individual has a GA, a black and a green peak, and the patient here is having a TT. So this is insertion deletion mutation homozygous which was present. So the first thing that we do always is that we look for if there is a mouse model available for knockout of this gene. And when we looked at that time, a mouse model was available, and in the mouse what was seen was a similar types of abnormalities in the mouse claws, and it was shown that the gene is expressed only in the hands and feet of the developing mouse. Okay? So this was really interesting. We thought we are really onto the track to identify this as the novel gene which causes the disease. And then when a series of experiments we did, because this is a transcription factor, and we could ultimately show that. So this gene is also very interesting. We see so many of these interesting things in genetics. You can see here point mutations in this gene cause a disease called as mesoaxial synostotic syndactylity. It is basically 4 fingers. The 2 fingers are joined in the X-ray, you can see. So these people have 4 fingers. On the other hand, if you have a duplication of this gene, we get a condition called as ectrodactyly, that means separation of the fingers. So you have on one side a point mutation causes syndactyly, on the other side the duplication causes ectrodactyly or separation of the fingers. This was our patient, and we could show that the mutation that we had found was much different than what these other patients have, which led to this particular condition. So this was our first gene that we reported as a novel gene for any disease from India for a condition not known yet. And after that, the NGS sequencing has come into routine practice. Now every day we do exome sequencing, genome sequencing. It has applications in rare diseases, it has applications in molecular oncology, in identifying pathogenic organism. We have seen in COVID pandemic, it has a lot of applications to sequence the genomes of bacteria, virus, and humans. And so it's no longer now a really new technology, it is how we use it in our practice as well as in research. So this was the first part, that is the how a book of life is formed and how we go about identifying these mutations. That brings me to the second story, that is the letter G or guanine. And guanine, the story's title is Goldsmith's Dilemma. Now all of you, I suppose you must have gone to a gold shop. So when somebody comes to a gold shop and he wants to sell his ornament, okay? So we can see at all these ornaments by looking, can we say whether they are real or fake? Okay? For us it does not matter, but for the goldsmith it matters a lot whether it is real or fake. So when someone comes to sell him an ornament, he is going to use different strategies. He is going to first visualize and see with his experience, is it real? He may use some sort of a carat meter to identify is it real or not, and then maybe he'll do a final test, that is a purity analysis by melting, because it is important for him. Same for a geneticist, whenever we find a variant, it is very important for us to identify is it a benign polymorphism or is it a disease causing mutation, because the patient's management, prenatal diagnosis, counseling, everything is depending on that one report that we give. And as I told you, whenever we sequence, if we sequence all of us, we are going to be 99.9% same in the genome. These are what we call polymorphisms. These are all what make us different from each other, but they don't lead to any disease. So whenever we find a variant, we always have to differentiate between a disease causing pathogenic variant and a benign non-disease causing variant. And we use different strategies for doing this kind of analysis. Let us see using one particular example here. So if we take this patient who came to us with coarse faces, with the x-rays showing this typical pattern called as dysostosis multiplex. So looking at this particular patient's clinical features, the corneal clouding, coarse faces, normal IQ, we had a working diagnosis of Maruti-Lamy syndrome. We did the enzyme assay is deficient. So we have a clinical characterization, that's a Maruti-Lamy syndrome. The next we go on to the molecular characterization. We identify what is the genetic mutation that is leading to the disease. So when we do the sequencing, we find that there is one change G2A in the patient. But how do we know that this is the cause of disease? Because it might just be a benign polymorphism. So there begins the search whether it is a disease causing variant or not. The first step that we use is a in silico characterization. So we use computational models, pathogenicity, the likelihood of pathogenicity, whether it is likely to be disease causing or not. Different tools are available for predicting the pathogenicity. That is one evidence we use. The next we go on to use some sort of a protein structure characterization. So we make the protein structure in silico, change the particular amino acid, and then see what is the effect of that amino acid on the structure of the protein. And from there we can predict maybe it is disease causing or not. So this is one more evidence that we will use. However, the final evidence always comes from the functional analysis, because all these are in silico, they might be wrong. So for a functional characterization, we will take the whole blood, the normal blood, we will extract the RNA. From that RNA we will make cDNA, and now in this cDNA we introduce this cDNA into a normal cell. Okay, so this cDNA is overexpressed in a normal cell. So when you overexpress it, you will get a huge amount of enzyme activity. So the normal activity in cell is 140. When you overexpress, it goes to about 1000. Now we introduce this mutation what we found in our patient into that cDNA. once you introduce that mutation, if that mutation is non-disease causing, we should have the same 1000 activity. But what we see is that when we introduce this particular mutation, the activity comes down to the baseline activity, which suggests that this is really a disease causing mutation. That's the final verdict which says that this is a disease causing mutation. So these functional assays help us to identify whether this is a disease causing mutation or not. Once it is identified, we report it to databases like ClinVar, so that second time when somebody identifies that same mutation, you don't have to do all this. You can directly report it as a disease causing mutation. Now this we would do for usually the mutations or genes which are known, but always we get unknown mutations or novel genes. Now novel genes, this kind of functional assay is not enough. So for that then we will go on to different animal models like the worm, the C. elegans, the Drosophila, the zebrafish, the mouse, or even the chimpanzee. And now what we recently use is something called as induced pluripotent stem cells. You can take the skin biopsy, take fibroblast of the patient, put it in the lab, convert it into a stem cell, and then that stem cell can be then be converted into a neuron or a cardiac cell or a muscle cell. Here you can see the stem cell has been converted into a cardiac cell, and then it starts beating, because that's the job of the cardiac cell. So now we have this technology to be able to look at that patient's cell by converting into, we cannot directly take a brain biopsy of the patient or a cardiac biopsy, but we can take a skin biopsy and then convert it into a brain cell or a cardiac cell, and then do whatever experiments you want to do. If you want to develop a new therapy, if you want to see whether a new therapy works or not, all this is possible. So that's about going about finding what is the pathogenicity of the variant. That brings us to the third story, that is the C letters signifying cytosine. And here we talk about how we can, so once you have identified the variant, you have decided it is disease causing, the next question the patient is going to ask you, can you do something about it? So what can we do? So the typical medical paradigm is that the patient gets sick, goes to the doctor, treatment is given, and patient is happy. Usually it doesn't happen so in the genetic diseases, because many of them we do not yet have therapies. We can only give them supportive treatment, but what we give all of them is something called as genetic counseling. And what constitutes genetic counseling is basically informing the family what is the disease that is there in their child, what are the complications expected, can we reduce those complications, is there some novel therapy coming up, can this disease be prevented in the next child, can we do prenatal diagnosis, what are the other options. When you give information to the family, that is what constitutes genetic counseling, and this we are able to offer to every patient that we come, although we may not offer a curative treatment. So there are a lot of misconceptions about genetic diseases which need to be sorted out. Like people think genetic diseases are rare, but as I told you, there are more than 7 crore patients of genetic diseases in India. We feel genetic diseases are always inherited and that family history should be present, but we now know that there are so many de novo mutations where there will be no family history, but suddenly a genetic disease can come. We think that genetic treatable, which was true few years back, but now we have so many newer and newer therapies coming up that we really cannot say so. And so there is surely hope for these patients because of so many different kinds of therapies that are coming up. So if we look at the therapy at the level of mutant gene, these are the gene therapies. We already have gene therapies for certain conditions. We can treat at the level of the mutant RNA, at the level of protein by replacing the protein, something called as enzyme replacement therapy. Every day patients come to us for enzyme replacement therapy. We have metabolic function can be deranged, so that can be corrected by dietary therapy using special diets, especially in amino acid disorders. We give the amino acid that is deficient in that diet, like in phenylketonuria. or we can treat the clinical phenotype through surgical therapy, as we saw in the earlier patient, by plastic surgery, or by a transplant, a kidney transplant or a liver transplant. And all the families, we can always offer genetic counseling. So at different levels, these therapies are available for some of these diseases, and that we do give. Let's see one particular case where novel kind of therapy we could give to the patient. So this was a patient who came to us, and so this patient had something what we call is a paroxysmal dyskinesias. You can see this whole day this patient would have this kind of movements. So because of this abnormal movements that they are not under his control, even during his sleep he used to get bouts of these movements. These are called as paroxysmal dyskinesias. So when this patient came to us and we did the exome sequencing, what we found is that this patient had a mutation in one gene called as ADCY5. ADCY5 is a gene, it's an adenyl cyclase gene. It creates cyclic AMP in the cells. So what is the mechanism is that this particular gene, so we have the adenyl cyclase enzyme which leads to cyclic AMP production, and it is activated by a receptor called as A2A receptor, and the ligand is this adenosine. Okay. So what happens when there is this particular mutation? It's a activating or gain of function mutation. So when this mutation is present, whenever adenosine comes, this receptor gets activated, the adenyl cyclase gets super activated because of this gain of function mutation. Lot of these neurotransmitters are produced, and that is leading to this abnormal movement in this particular patient. Now what happened serendipitously is that this patient, one of these patients in the country Madagascar, he went to his doctor and he told his doctor, I have all these movements throughout the day, but when I go and drink coffee, my movements stop. Okay. So this was very intriguing to the doctors. They thought he is putting just 2 and 2 together. This might be an association. They did a double-blind study. They admitted the patient and they gave him coffee, and the movements totally would stop. And then when they withdrew the coffee treatment, the movements would come back again. Okay. So that was very surprising. What was noticed. So they gave them decaf coffee, that is coffee without caffeine, and the movements did not subside. Okay. So what was happening is that caffeine structure, so when you give caffeine, it dislodges adenosine and binds in place of adenosine and inhibits this receptor, and the movements come down. So this was a very serendipitous kind of a thing. So when we read about this, we also gave our patient coffee, but coffee, how do you give? He needed to take this whole one packet at a time. Okay, and our coffee has this chicory, it's a very bitter thing. Okay, so we had to import caffeine tablets for him after some time because he could not really take this coffee. But what was noticed was very dramatic, that you can see this same patient now, and what has changed is that his movements went off totally, and he is now able to lead really a normal life, doing cycling, doing swimming, what he could not do earlier. And so earlier he used to get about 20 events in a night, and now they have gone to less than 2 events per night. And so you can see this dramatic change that is not really more of a big kind of therapy. It is just serendipitous observation by some clinicians and then putting it to use. And still now he is on follow-up with us, leading a really normal life. So this was really a nice experience for all of us to make use of something routine as coffee to use for treatment of this patient. So that brings me to the last story, that is thiamine. And this story tells us about how our traditional medicine is meeting the modern medicine. So in our traditional medicine, we have the traditional Ayurvedic medicine, where the principle is that the Ved will check the patient, and individually the Ved is going to make the treatment for this patient and give it to him. Whereas in the modern medicine, what we have is one drug for all patients. So all patients with fever get paracetamol, all patients with malaria get chloroquine. But what it creates is that all patients do not respond in the same way. So the current practice is a one size fits all or a trial and error method. But what we are going towards is something called as personalized medicine, that is right treatment for the right person at the right time. Okay? And this is classically shown by this example of a drug called as 6-mercaptopurine. This is a drug routinely used in treatment of leukemia in children, blood cancer in children. So what happens when you give this drug? There is an enzyme in our body called as thiopurine methyltransferase. This enzyme breaks down the drug and it is excreted in urine. Now in population what happens? 88% of the people have this normal enzyme activity, 11% have intermediate activity, and about 1% have no activity of this enzyme. So what happens when you give drug to 100 people? 88% of people will respond to the therapy and they will excrete the drug in urine, everything is fine. The yellow ones are the 11% people who have less activity of this particular enzyme. So their drug will remain in their system for a long time. So they will respond to the therapy, but they will get lot of side effects of the drug because the drug is in their system for a long time. And unfortunately, this one person who has no activity of the enzyme, he will die because of the medicine instead of the disease. So what can we do? If we can do a test and identify these 11 people and this one person, then we give low dose of drug to these 11 people and don't give that drug to this one person, and all 100 people are happy. And that is what is the principle of pharmacogenomics or pharmacogenetics. And this is seen with all different drugs, whether it is hypertension, heart failure, antidepressant. We see that all patients don't respond to the same drug in the same way. And so instead of going to a reactive kind of medical care that we have at present, where we have the diagnosis, give one drug, not working, change the drug, and the patient is in disease for a long period of time. Instead of that, what we are moving towards is something called as a personalized medicine, where at the time of diagnosis, you do a test and find out whether that person is likely to respond to that drug properly or not. Give the right drug and patient is in disease for a much lesser time. Even going further, we have the field of predictive medicine, where now we are trying to find which are the people who are more susceptible to develop a particular disease in future, and try to put in some preventive aspects to even prevent that disease. Okay? So from treatment to now we are going towards preventing the lifestyle diseases or delaying the occurrence of these lifestyle diseases. And this is not really science fiction. We already have multiple tests already available. In USA, if you don't do a test before giving carbamazepine, it becomes a medical negligence, because few people given carbamazepine can develop a severe condition called as Stevens-Johnson syndrome. We still don't have such regulation in India, but it's surely going to come in future. And for the future clinicians, it is very important to keep this in mind, that in future you are going to treat patients based on the knowledge of what they are susceptible or not susceptible to. So in future, probably when a child is born, we are going to have the blood sample of the child to do the genome sequencing, the proteome, the metabolome, put that everything in a chip or a card, so that whenever you go to the patient, to the doctor puts the card in the computer and gets to know what you are susceptible to, what you are not susceptible to, which drug will be best for you. And so instead of the horoscope, we might have something called a genetic horoscope or gene patrika. And again, this is not really science fiction. In Hyderabad, we have already a company who makes this genome patris. But the problem is here that we still don't understand properly about this aspect of genetics. The predictive medicine, still we learn a lot before we really implement this in practice. So that was about the whole world of genomics that I wanted to introduce to you. I'll briefly take you through what is happening in India today from the genomic landscape, and through the different aspects, that is diagnostics, education, the databases, some of our research, and some of the policies and guidelines. If you look at the genetic diagnostics in India, we started just like any other country. We started in the initial days by doing clinical syndrome diagnosis, imaging, dysmorphology databases, and slowly we moved on to cytogenetics, FISH, Sanger sequencing, MLPA in the early 2000s, and then towards microarray, exome sequencing, genome sequencing, and the most latest, the optical genome mapping. So what I want to show you is that now we don't really have any excuse saying that something is not available in India. Every diagnostic tool or facility, we have it available in our country, including the most latest optical genome mapping, which is a very, very high resolution kind of a karyotyping. You can look for the chromosomes at the DNA level, and especially for a disease called as FSHD, facioscapulohumeral muscular dystrophy. That's the only diagnostic test that is available. Till about 4 years back, we had to send the samples abroad for this only one test. Now that also is available in our country. We have so many centers across the country who are able to do all these diagnostic tests for different genetic diseases. Early diagnosis is important, but judicious use of these technologies is important, especially because many of them are quite expensive. From the education point of view, we have different educational programs available. The DM program in medical genetics, which is a medical program. We have short duration courses. We have a publication which comes from Genetic Clinics, a 3-monthly publication from the Indian Academy of Medical Genetics. And especially I want to introduce you to this online training program called as Gene Top. Okay, this is very important program, a free program created of 57 lectures on different basic aspects of genetics and genomics, on clinical genetics, simple lectures like approach to a child with short stature, approach to a child with intellectual disability, what tests to do. We have all these lectures freely available. More than 3,000 clinicians have registered and gone through this whole course. I hope some of you at least go through this course. It has a lot of material available, again freely available to everyone. Coming to next, the databases. I told you that we need to know what are the polymorphisms in a particular population. So what databases we have till now are all the Caucasian databases, the Nomad, the Exact, the 1000 Genome. These are all Caucasian databases. They don't help us much in our analysis. We had very small Indian databases, and that is when we went on to create our own exome database of 1400 exomes. This was our initiative in 2021 because we needed this. Without those databases, it was very difficult for us to analyze the exome and the genome sequencing data. And this was now freely available through our website on CDFD. And the recent one is the Genome India database, which is a database released on 9 January 25 by Prime Minister of India. It's a 10,000 Indian population whole genome sequencing of normal individuals. This is really a gold mine of data that is available now of 10,000 normal human genomes, and it is going to throw out a lot of challenges, lot of research activities that can be taken up using this data. Regarding the policies and guidelines, so one of the societies is the Indian Academy of Medical Genetics. I told you this Gene Top is one of the activities. Similarly, we have a lot of guidelines and position statements that have been given through this society, like prevention of discrimination based on the genetic information. Also, the prevention of direct to consumer tests like predictive medicine, which are not to be used, or carrier screening in couples, which tests should be done for carrier screening in couples, and more guidelines are yet to come. The next aspect is about the research that we are involved in recently, and that is the program that Dr. Alex spoke about, is the Mission Program in Pediatric Rare Genetic Disorders. This is really an ambitious program that we took on about 2 years back, where we are collaborating with 16 different centers across the country to recruit patients with rare diseases. Once they are recruited, they send to us for exome genome sequencing. If we identify a cause, we provide counseling to these families. If we don't know, then we identify the variants, and then we have a number of basic scientists who are collaborating with us. They make use of Drosophila, zebrafish and mouse models to identify the novel genes. We have a collaborator in CMC Vellore who is helping us with some lentiviral vectors for therapy development, and we have a number of programs for awareness. So it's really a good collaborative project where we have put together the clinical centers across the country and the basic scientists in our institute as well as in 2 other institutes to come together and work. And I'll just show you quickly 2 examples of what we have been able to achieve in this program. This is one particular novel gene discovery program that we did, where we had these 2 families who came to us with spastic quadriparesis, that is neck down, these children were paralyzed from birth. And when we did exome sequencing, we could identify in both these families a homozygous mutation in a gene called as AIMP2. Now nothing is known about this particular gene AIMP2, and at that time we have just reported that this is a stop codon variant in a novel gene for this particular disease. So when we started this program, we saw that this particular complex is conserved in Drosophila. So one of our collaborator said that we will model this gene into Drosophila, that is the fruit fly. Okay, so they went ahead and did a CRISPR-Cas9 based knockdown. They removed this particular gene from the Drosophila, and then they could show that these knockdown flies, they were viable but infertile, and they could show in the brain. So you can see here, this is the expression of AIMP2 in the neural stem cells and the neuron and the glia. You can see that there is a lot of expression in the normal brain, but in the mutant the expression is reduced, which again points to our what we saw in the patients, a neurological phenotype. Next, they went on to see the head size, but they could see that there is no change in the head size of the Drosophila flies when this mutation is present. But when they looked at the behavior assays, one is the flight assay, they could see a drastic reduction in the flight assay and in the climbing defect in these flies. So using these experiments, they could show that AIMP2 localizes to the neurons and the glia, that these animals survive but they have behavior alterations. And you can see this very nicely in these videos here. So if you can see here, these are the 2 flies. So these are fruit flies. This is the normal fly. If you give a puff of air to this fly, you can see that it starts fluttering its wings. But the wings you can't even see, it's fluttering so fast that you can't see those wings visually when the fly is fluttering. On the other hand, this is the mutant fly. When we give a puff of air, you can see that it is starting to flutter but very slowly, and even when it is fluttering the wings, you can still see the wings of the fly which are not seen. So this is one motor defect that is visualized. And the second is when these flies are put in a test tube, they always have a tendency to move upwards. So the first 3 test tubes are the normal flies, and the 4th one is the mutant fly. And you can see that when this test tube they are put, the first 3 test tubes, the flies are able to move upwards more than 50%. In the last test tube, these flies, the mutant ones, they are not able to climb above the 50% mark. So these are some of the ways in which we try to model these diseases in animal models to show that these are really the disease causing genes. Here is another example. We had another family who came to us with 2 fetuses who had hydrops fetalis. That means there is lot of fluid in the around the heart and the lungs. So this family had 2 fetuses who were affected with this hydrops fetalis, and when the fetus was aborted, we found that there was no much morphological abnormality, but what we could see is that there were a lot of these blebs that you can see around different organs, the heart, the liver and the kidney. So this was very surprising. What are these blebs? This family went on to have another child, and the second fetus also had the same type of blebs. And when we did the histochemistry, we could see that there is expansion of the extracellular matrix here in these blebs. So we did the exome sequencing and we found a stop codon mutation in this one gene called as SERPIN A11. So SERPIN A11, SERPINs are serine protease inhibitors. There was nothing known about this particular SERPIN A11. It was a non-characterized protein. But SERPIN A1 is a very common gene known, because alpha-1 antitrypsin deficiency is a very common disease that is known. So since nothing was known about this particular gene, we went ahead with the functional characterization. We made first the model using the alpha fold, and we could see that this pink colored part of the protein that is lost in the mutant protein because of this stop codon mutation here. Next, we made an antibody against this particular protein, a commercial antibody, and doing a Western blot, we could show that there is total absence of this particular protein when there is a mutation. And we looked at expression of this protein in different mouse tissues, and what we could see that this protein is expressed in almost all tissues, including lung, brain, liver of the mouse. So we do a histochemistry, a immunohistochemistry, and you can see here these are the mouse bronchioles, where the green colored you can see this is the SERPIN A11 prominently expressed in the mouse bronchioles, not much in the liver and the kidney. And same thing when we did in the fetus, you can see that this is the normal fetus with the normal fetal bronchiole, and this is the fetus with the mutation where the expression is almost not there. Then we went on to study the RNA in different tissues of the fetus, and we did a transcriptome, and that also showed that there is a severe reduction of transcription for this gene. So overall, we could publish this recently in 24 as a novel serpinopathy, another novel gene added to the already known genes. And now we are in the process of making a mouse model. The heterozygotes are already there to study this gene further, so that we see what this gene causes when there are mutations in them. So overall, that's what is the landscape of genomics in India, where we have come from doing only simple tests like a chromosomal analysis in 1980s and 90s to now doing almost the novel gene identification. When I was doing my DM, we used to imagine and dream that maybe someday we will also be able to do what we were reading in the journals. And I am so happy now to be associated with this institute, where we have so many of our basic scientist collaborators who help us to do the further work that we are not able to do. We have so many clinical collaborators throughout the country. We have such rich availability of patients whom we can help by doing this research. And so the acknowledgements go to all these families of patients who provide their samples to us, so that we can work on them, so that future patients benefit from whatever work we do. And that's our dynamic group at CDFD. Many of you who came to our institute must have met many of them. They are young scientists who are very dedicated to the work that we do. We have, this is the group of the mission program. We had a meeting when all of us came together. And so really where we have all come by sitting on the shoulders of the giants who did all the work earlier. Okay, and so we can see forward, because some amount of work has been done by our predecessors. We add to that work, and I hope that the future scientists sitting here will add more. It's an incremental, science is always incremental. So hope I have provided you enough data, enough information to excite at least some of you to jump into this field of genomics and feel the same excitement that I feel every day when I go to the lab, that I'll get to learn something new. Thank you very much.
Thank you, sir. Thank you for providing us with a deep understanding of current state of genomics in India and the exciting possibilities that lie ahead. Thank you once again, sir. I request you to kindly stay back for the Memento presentation. I request the other dignitaries to kindly join in with the orator on the dais. We also have our CEO, Dr. Benny Joseph, with us. Sir, I request you to kindly join there. I request Mrs. Devi, wife of Dr. D.M. Vasudevan, to join the dignitaries on the dais. I request Reverend Father Terin Mullakara, Assistant Director of Medical Education and Services, Jubilee, to hand over the memento to our esteemed speaker as a token of love and appreciation. I also request Dr. Benny Joseph, our CEO, to present a gift to our orator as token of gratitude. Now I request Dr. D.M. Vasudevan to present the book written in his honor to Dr. Ashwin Dalal. I also request Dr. Vasudevan sir to sign an autograph on the book. This book is written by Dr. Kesia Kuruvela. Once again, thank you all. I request Vaswan sir to stay back and request other dignitaries to join the audience. There is a special announcement that will be made by our research director.
Dear friends, the official function is over, and I'm very glad that this has been conducted in a very orderly manner, and thank you very much for all my colleagues to conduct it in a very grand fashion. And thanks for our guest. This is only to state one announcement. That is, see, we have PhD scholars are here. This institution is recognized by the university, our university, that is the Health University, Kerala Health University, for the PhD. But that PhD is only for medical graduates. That was the problem. And now, this year onwards, we are starting the PhDs for the non-medicals also. That is in connection or in concordance with the Manipal University. With that, I thank you all the people that came here. Thank you very much.
Thank you, sir. Yes, JCMR is now recognized research center of Mahi under the Faculty of Life Sciences. From now onwards, we will be initiating PhD program of Mahi in Life Science at JCMR. The research areas will include biology, biochemistry, cell and molecular biology, cytogenetics and genomics, disease biology, drug discovery, metabolic disorders, phytochemistry, and structural bioinformatics. Thank you. Now I invite Dr. Matthew John, scientist, JCMR, for the vote of thanks.
First of all, let me thank Almighty for again making the 7th DM Endowment Vasudevan Oration a grand success. Let me come to my duty. My first thanks goes to today's beloved speaker, Dr. Ashwin Dalal, for a wonderful talk. Sir, you conquered our heart and brain equally. It was a fusion of genetic ragas using the 4 letters A, G, C, T. Sir, it was a treat to watch, and we witnessed a fantastic oration. Thank you, sir. And The next thanks goes to our ever beloved Vasudevan sir. With great honor and respect, I owe my vote of thanks on behalf of entire Jubilee family for your presence itself and making all of us blessed during this event. And because of you, this event is made possible, as Varghese sir told. Thank you, sir. And along with sir's integral component in life journey, Devi madam also came here, and thanks to madam also. And next thanks goes to our ever-loving, charming director, Father Reni Muntan Kurian, who is always deeply passionate and enthusiastic about research and promoting the research to the core and a pillar of our strength. And we are blessed with your presence also. Thank you, Father. And our newly joined Assistant Director, Father Terin, also is here. He is also really interested in research and asking curiously about the happenings of this program yesterday also, and was tracking and monitoring all the events with good keen interest. Thank you, Father. Next thanks goes to our principal, Dr. Praveen Lal Kuttichira. Sir has delivered a deeply devoted thanks to Vasudevan sir. It was always a great treat to watch the disciple giving the respect to the teacher. And this happened because of Praveen Lal sir. Thank you, sir, for your presence. And then we always used to say, and we used to see in a lot of team meetings, that a captain should lead from the front. And we have a true leader in Dr. P.R. Varghese, who has been our great mentor. And thank you, sir, for your presence. And next thanks goes to our beloved Dr. Benny Joseph, our CEO. Sir is always a truly professional man, always being actively involved in our research activity and promoting research to the core. And we are happy to have your presence here, sir. Thank you. Next, my thanks goes to my colleague, my loving colleague, Dr. Alex George, for introducing with all professional concepts the speaker for the day, Dr. Ashwin Dalal. Thank you, Dr. Alex George. And then we have our medical superintendent, who is always ever charming fellow. We enthuse this positive energy from him with his ever gracing smile. And thank you for your presence here also. Thank you, sir. Thank you. And thanks to all the vice principals, to the two vice principals we have, Dr. Padma Kumar and Dr. Cherish Paul. Your presence also made this event a very special one. Thank you, sirs. And next, my thanks goes to the backbone of this program, the entire Jubilee Center for Medical Research family, the JCMR family. Special thanks to my colleagues, Dr. Suresh and Dr. Alex, and all one. I'm worried really whether I could spell it out every name, so I'm just not mentioning everyone. To the core, they worked day in and out for making this happen. And thank you all, JCMR family. And the next thanks goes to the administration team, who has been a pillar of support for making this event a grand success. To every program, they have cooperated with us. And thanks to the administration team and the finance team, the purchase department, the engineering and maintenance department, the canteen service facility, the hospitality from the guest house, a lot of people involved. And thanks to one and all, and thanks to the entire Jubilee family for making this event a grand success. Thank you one and again all. Signing off.
Thank you, sir. Thank you once again to one and all. Please join us for the tea, which will be served to your left side of the auditorium. After the tea, we'll be having discussion with the orator at New CCM Hall. Kindly join us after 15 minutes. Thank you so much. Take care until we meet next year on 7th March 2026 for the 8th discourse of Dr. D.M. Vasudevan Endowment Oration. Thank you so much once again.